All right, we left off on replication, and the one thing we didn't get to was how do the ends get replicated. And I alluded to this before when we were talking about structure of chromosomes, but we need to talk about the telomeres and how the telomeres get reproduced. So here's the slide of the telomeric ends. Now this is abbreviated, right? This isn't quite factual of how the telomeres are, because if you remember the two loops where the telomeres are kind of looking like it's a circular chromosome, the 3' end is actually much, much longer than this, because it loops all the way around and makes a lot of base pairs with the other end. So this is just an illustration for the purposes of keeping it simple on here. But the telomeric ends has got some of the 3' is single-stranded, and it's longer than the 5' end of it. Now this is greatly simplified, but the theory stands. When you try and replicate the ends of these, you have to remember that DNA replication is semi-conservative, right? So the two strands are stuck together here. When they get pulled apart, one strand is then the template for the other, and then the other template is the one for the other. So if I pull these apart, I have lost this information if I'm going to be replicating that strand, right? I separate these two out, and when I go to replicate and copy that strand, I've lost the information that's supposed to be there in the single-stranded part. So anytime DNA is single-stranded and doesn't match up, then this is a loss of information, right? So if I were to replicate this, I'll have lost this overhang. Now, cells can deal with that. You lose the overhang, they just make the rest of it single-stranded, and the telomeres are really long, repetitive region, right? So if I lose a couple of repeats, no big deal. I just make more of it single-stranded. It kind of chews back a little bit, but you can still form the same looping structures at the end, okay? But every time you replicate, every time that cell divides, I'm losing a piece there, all right? When the other thing too is on this strand, as this is the template strand, if I'm replicating that, the first thing that comes in is the eukaryotic equivalent of primase, right? We're talking in eukaryotes. We separate the two strands. That polymerase that comes in that acts like primase puts down an RNA primer first, and then you extend the rest of it. That RNA primer is not a long-term portion of the chromosome. So that gets eaten away, and you're going to have the gap left by primase, right? So every time I do that, that's shortening the other strand too. Both strands are shortening every time you undergo replication, okay? Now, that's fine for a cell who's only going to divide a certain number of times. You've got long telomeric repeats. You can shorten them after a while. The cell recognizes when the telomeres are getting too short, though, and it won't divide if the telomere is too short. It says, this telomere is too short. I'm going to start encroaching in on coding information, and so it acts as a clock for a cell. Cells will stop dividing if their telomeres are too short. So there are some cells, though, that actually continue to replicate indefinitely. The cells will continue to divide. There is a group of cells, they're called HeLa cells, H-E-L-A, and they were originally isolated from a woman named Henrietta Lacks. They're cancer, I think they're cervical cancer cells, so they removed cancer cells from her and started growing them in culture. We've been growing them in culture since the 1950s, right? Her cells are still alive in thousands of labs all across the world. Companies grow these and actually sell them. You can go buy HeLa cells. There's an interesting book that's come out that says, it's called The Immortal Life of Henrietta Lacks. And it talks about kind of ethics of, whose cells are those? Should her family be getting all the royalties and all the profit from all the cells and all the research that's been done with those cells? Or, are cells cells and they're no longer owned by the person who donated them? It's an interesting book that talks about stem cells, cell culture, and ethics of biology. The Immortal Life of Henrietta Lacks. In those cells, though, that can continue to replicate, it's because they can extend these telomeres, and they no longer have that clock saying that they're old cells. So we've passaged those cells hundreds of thousands of times, and the telomeres are still the same length. That's because of an enzyme called telomerase. And what telomerase does is it brings in its own primer. It's got its own piece of RNA that's going to act as a primer, and it's going to extend that long 3' end, okay? I can't extend this long 3' end here by a traditional polymerase, because it doesn't know what to read, right? There's no other strand. There's no complementary strand out here for me to extend that, right? Even if a polymerase could put nucleotides there, it doesn't know what nucleotides to put, right? So what telomerase does is it comes in and it has its own RNA primer. Here's just an abbreviation of the RNA primer. Remember, the telomeric ends are repetitive. So even though there's not a template strand there, if it's repetitive, we know what the repeats should be. And so the repeats are dictated by that primer, that RNA primer that telomerase is bringing in. This yellow floaty thing is the telomerase enzyme itself. It's a polymerase, and it's a polymerase that reads its own template. It's just like talking to itself, kind of, right? It knows what it's going to say, puts it down, and then it says it, right? So it comes in, and it's going to actually extend and act as a polymerase and stick nucleotides there. Then it lets go. It can sit back down again, and it can base pair with the repeats again, because it's just saying the same thing over and over again, just repetitive things. So it can lift off, bind again to that same end, and keep extending that end. So you just keep going and going and going. Right, so it put those down, lifts up, binds to those, it extends it more. It could get off, shift down, extend it more. And it could keep making those repeat ends longer and longer and longer. This gives the cell immortal life. It can continue to divide as many times as it needs to.
Why does it attach where it attaches and not closer to the other? Oh, I see.
So it's just base pairing to a couple here. It can now extend this region because it knows what the sequence should be, right? So it's filling that in. Now once it fills it in and puts another, it'll put another T, T, G here, well then the whole thing could lift up and this binds to TTG. So it could bind again to here, extend it longer. Lift off, bind again, extend it longer. Do that over and over again. Yeah?
I'm wondering, how does it know when to end?
That's a good question. I don't know. Somehow the cell knows how long those telomeric ends need to be, and it'll only extend it as much as it needs to. But I don't know. There's nothing coding for it in the telomere, right? I mean, you could just keep doing that forever and ever and making a huge long chromosome that was all repetitive. Somehow the cell knows how long that's supposed to be and stops. So these immortal cell lines, you have some cells in your body that are expressing telomerase, notably your germline cells. So cells, oogonia and spermatogonia, the ones that are dividing to become eggs and sperm, those are immortalized. They're kind of stem cell nature-like. In almost every single one of your tissues, you've got some cells that are set aside to be replacement if you've lost or damaged cells. So almost all your adult tissues have a couple of cells that are stem cell-like, and those have telomerase turned on. And then if you have a cancer, and not just a benign tumor, but an actual metastasized cancer, those have probably immortalized. That's what's allowing them to grow huge and out of control. Okay. Questions on telomerase? This is one of the commonly upregulated genes in cancer cells, right? If a cell is not cancerous, it doesn't have telomerase turned on. If you've got telomerase turned on in a cell inappropriately, it's usually a sign that it's a cancerous cell. So there's some chemotherapies, they're not quite chemotherapies, but cancer treatments that are trying to kill cells that just have telomerase turned on. Now that's also going to kill off germline cells and stem cells as well. So you have to be a little bit more specific about what kind of cell that has telomerase turned on. But that's a good first marker of like, where's cancer going on in your body? Well, let's just isolate all the cells that have got telomerase turned on. So, Victoria, do you have another question?
Yeah, is the telomerase made in the nucleus, or is it?
Yeah, telomerase is just a gene that gets turned on. It's made in the nucleus, right? So most cells have shut off the region of the genome that codes for telomerase, and they're not expressing it. But cancer cells or germline cells, they've got that region of the genome open. They're expressing the telomerase gene. That turns into protein and extends these telomeres. So yeah, it's on the nuclear genome. OK, so that finishes replication. That's how you replicate the ends. We're going to talk about PCR. PCR is replication, but it's in a test tube. And what's cool about PCR, so it stands for polymerase chain reaction. So we're using a polymerase, And the cool thing about it is that it's a chain reaction, that once you start, you can keep amplifying lots and lots of copies. So you can start with a tiny amount of DNA, and you can make lots and lots of copies of not only the entire thing. We could throw in some random primers that would just stick down anywhere on our piece of DNA. If you got some DNA in a test tube, you could throw in random primers, and they would just bind at all kinds of random places. Throw some polymerases in there, and you could get the whole thing to replicate, right? What's cool about PCR is that it's specified. We can determine exactly the piece that you want to make lots and lots of copies of and not bother with anything else. So it's selective and it's amplificative. It amplifies, right? So how does this work? This was developed, like I said before, by Kerry Mullis in about 1983 when he was working at the Cetus Corporation. And the key to PCR is designing little primers, okay? When your cells are replicating, primase, or the eukaryotic equivalent, comes in and lays down the RNA primer, okay? And it does that at the origins of replication, right? Sits down there and makes those primers for you, or at the replication fork, right? What we're doing here is you can synthesize primers to be whatever sequence you want, okay? And we are just going to design primer on one side, primer on the other side, and that will define a region. And we'll say that's the only place that the polymerase is going to be active. Polymerases need a primer to start, and so if there's no primer, it's going to ignore all the rest of the DNA. So here in our example is the target sequence, okay? In this illustration, it'd be better if you could actually see the whole rest of the chromosome, but this could be a really long, intact chromosome. And what we're targeting it by is the way we have designed our primers. So you can take your DNA in the test tube, you boil it so that the two strands will separate, and we will design pieces of DNA that will stick, will base pair complement to whatever region of that chromosome you want. So this is showing you it binding to the target sequence. So this primer is going to sit down. I can throw polymerase in there, and I've defined the place where polymerase should start making the copy. And so it's just going to go in and fill in the new strand. And I'm doing replication in a test tube. You design a primer for the opposite strand. Polymerase comes in, and it's just going to make that piece. And it's chain reactive because we can just boil the samples again. Primers sit down again, and we just make another round of copies. So it's exponential growth, right? You can start off with a tiny, tiny fraction of DNA, and you can get hundreds of thousands of copies in just a few cycles of heating and cooling, heating and cooling. Okay? So I'm going to walk through each of these steps, and we'll talk about how you make primers. So you will get to design primers. So this is a little primer on primers. You will actually go in and design your own, so you'll get to know this intimately. But primers are usually about 18 to 22 bases of DNA nucleotides. Your cells use RNA, but then you have to chew away the primers. We're just going to start, and DNA nucleotides can work fine for a primer, and they will just get integrated right into your replication. There's no having to use a polymerase 1 kind of thing to chew away the primer and fill it in. Oh, just start with DNA primers. We can use a polymerase, and we don't have to chew anything away. This is synthesized DNA. Most of the time, you just have a company do this. There's several companies. You send them the sequence that you want them to make, and they just use their organic chemistry machines, and they synthesize the nucleotides in the sequence that you want. So they're pretty cheap, too. It's only about $1 per base. So for about $18, you could make a primer. $18 to $22, and they'll usually overnight it to you as well. You send it to them in the morning, they replicate it in the afternoon, ship it out overnight, it shows up the next morning, you get to do your PCR reaction. Organic synthesis has come a long way. We're pretty efficient at that. All right, so the first step is to denature your sample. All right, so you'll have to start with isolated DNA. And then you just boil your sample. The phosphodiester bonds in DNA are very, very stable. You can boil those, you're not going to break those. But what you're going to break is the hydrogen bonds. So you boil the sample for a couple of minutes at 95 degrees, and that's called the denaturing step. Okay? Got single-stranded DNA. If you keep it at a high temperature like that, they'll stay separated. As soon as you start cooling that sample down, the DNA is going to base pair together again. So we have to keep temperature very, very precise in doing PCR to make sure things stick together when we want them to and things stay denatured when we want them to. So the two strands will separate. And then right in that tube, you've just put your primers, your small little 18 to 22 base pair primers. You're going to make two different primers, right? The two that flank the side that you want to amplify. So there'll be two different sequences. You can just throw those both in the same tube, boil them. Those will denature too. And then you cool it down somewhere between 50 and 60 degrees Celsius is usually the sweet spot. And what that's going to do is allow your primers to specifically hydrogen bond to the template strand. Okay? And this is based on calculations on the GC content, and you kind of estimate how tightly are your primers going to stick. So depending on the primers you make and how long they are, you're going to keep them somewhere in there. If you made a really, really long primer, it would probably stick really well. So you could still keep these temperatures kind of high, 60, 62, 65. If you made a really, really short piece of primer, then you probably have to cool it down a little bit more to get it to stick, right? So the sweet spot is kind of between 50 and 60. So the primers stick. They just base pair. And they're only going to stick to the complementary sequence. Amazingly enough, 18 nucleotides will get you to almost only one place in the entire genome. Right? You can design 18 nucleotides, and there's some repetitive regions. So if you made a repetitive one, it might stick to a bunch of places. But the DNA code is actually very, very dense. So 18 nucleotides will probably only sit down on one location in your entire genome. Especially if you pair it up with another 18 base pairs, those two sitting next to each other, you're probably only going to amplify one place in the entire genome. So you can just isolate entire genomic DNA and amplify whatever specific region you want. Now, this requires you to know something about the sequence, right? So you already have to have sequenced and know what the base pairs are in order to design your primers. But there's so many genomes sequenced now, that's usually not a problem, right? Unless you're working with something really obscure. All right. So the first step was denaturing at 95 degrees. Second was annealing, allowing those to cool down and base pair. The third step is extension. Here we're going to heat the sample up just a little bit. And we do this because this is the temperature that the polymerase is going to like to be active. Okay? Now, if we got DNA polymerase out of U, if in our test tube what we were using is human DNA polymerase, what temperature would it want to be at? What would be the ideal temperature for? What's your body temperature? 98.6. Yeah, 98.6, right? So that's around 37 degrees Celsius. Okay, so it would be much cooler. We're doing this, the extension for the polymerase that we use is actually 72 degrees. That's really hot. That's what is 72 in Fahrenheit? Now it's close to boiling, right? This is nice because what this is a thermostable polymerase. It's a polymerase that works at really high temperatures because we're going to use this polymerase and it's going to come in, it's going to sit down where the primer was, and it's going to extend. But the very next step is we're going to want to boil this sample again, take it up to 95 degrees. So we use a really, really heat-stable polymerase that will withstand that boiling temperature and still be active for the next round. So the extension is dependent upon the polymerase. I'll talk about the one that we use in just a minute. So you heat it to 72. The polymerase is going to synthesize a new strand. The only other thing you have to have around in this test tube is free nucleotides. Nucleotide triphosphates. The polymerase will do the rest of the work, right? It's just going to go down the strand. It's going to read whatever sequence is here. It's going to grab the appropriate nucleotide triphosphate, stick it in there, and you're going to have made synthetic DNA, basically. So in that first round, whatever strands that you had in your sample, whatever DNA you had in the tube, is going to be replicated. Okay? But then we've made a bunch of new red synthetic strands, new strands. So we boil it again, you separate all those, and now the primers, you put in a bunch of primers, so there's more primers that can sit down. You do the annealing step, they stick. You do the extension step again, you elongate, and we get more. You can just keep doing this over and over and over again. Typically, you do about 20 to 30 times. 20 to 30 times, you're just going to have exponential numbers of these guys going on, even if you start off with a tiny, tiny sample. This has allowed most of forensics to actually happen these days. When there's DNA evidence that's admitted into court, we can take tiny fragments of DNA found at a crime scene. You can get a swab of just a couple of cells. And by PCR, a couple of cells is enough DNA to amplify, and we can identify the sequence of that individual. We do single tadpole PCR in our lab. Just take a single tiny C-squirt. It's only about 2,600 cells. You throw it in there, extract the DNA. You can't even identify the DNA with any of the methods that we have. There's such vanishingly small amounts of DNA, we can't detect it. But by PCR, you can amplify it, and then we'll know the sequence. You can start off with just tiny amounts. So just a blood smear, just tiny bits of tissue or fluid. If it's got DNA in it, we could probably amplify it. So, which makes this really, really powerful. Before, if you wanted to sequence something, you had to have a lot of tissue and a lot of DNA in order to get enough to actually read it. Here, this amplificative process, we can just make a ton of it and then sequence it, see what's there. So, yeah.
I was reading in the book and it said something about like mistakes being more common in PCR.
Yeah.
Is it like a lot more common? Is it really hard? Because there's no proofreading, right? Are you gonna?
I'll tell you right now. It's a good question. The question is, mistakes. What about mistakes that get made? If you're trying to identify an individual, then if the polymerase that you're using is making mistakes, you're not really reading the sequence from the original sample. So we have to take that into consideration. The one that we've almost always used is an enzyme called Taq polymerase. Taq is the polymerase that comes from the single-celled bacteria called Thermus aquaticus. It means it lives in really high-temperature waters, right? Thermus being heat, aquaticus, water. So it's an aquatic bacteria that lives in like these steam vents, these geysers and stuff, like at Yellowstone, these really hot, almost close to boiling water. Bacteria actually can live in these environments. So one of these bacteria, if it can live there, then it's got a stable enough polymerase that you can heat it up to 95 degrees and not kill it. So it was named Science Magazine's Molecule of the Year in '89. I don't know how many other molecules of the year there have been, but bacteria are not so concerned about the fidelity of their sequence as you are, right? They're making a lot of them. And so this one actually doesn't have 3' to 5' exonuclease activity. It doesn't have that proofreading where it can back up one nucleotide and remove it if it's put a wrong one in. So if it puts a wrong one in, it doesn't care, it just keeps moving. And on average, this happens about every 9,000 base pairs. So now, each time it replicates, right? Every cycle in PCR, it might make a mistake here or there. You can often figure out which ones are mistakes and which ones are not, because not every single time is it going to make the same mistake. So if you do a couple of sequencing reactions, it'll kind of buffer it out. You can kind of weed those out a little bit. If you're doing really, really fine stuff that you don't want mutations in, we end up using a different polymerase. There's another one called PFU. I don't remember what the name of that organism actually is, but it's probably P is the beginning of its genus and FU is the beginning of its species. I don't know what the name of it is. It's got a DNA polymerase that does have proofreading activity. It's not quite as fast as Taq polymerase, and it's not as cheap. It's harder to come by. But it does have proofreading activity, and so it makes a mistake only about every 1.3 gigabases. So every 1.3 million bases instead of every 9,000. So much, much better fidelity. So if you really need something to be accurate, you use that guy. Yeah. Yeah, I don't know, actually. I imagine the forensics labs, in the sheriff's departments and things like that are using PFU. Yeah, I would imagine, or something else. Maybe there's a better one out. These were the two that were common when I was doing this a lot. So I never really cared so much, so I just used Taq and just figured if there's a mutation, I would do it a couple of times and buffer it out. So, all right, so you could take your same sample, you could do one PCR reaction and sequence that. If you did it like 3 more times and that sequenced all of them, probably not the same mutation is going to show up every time. So you just align those sequences, and the ones that are are variable, you could say, well, that's the wrong base. Like throw it out of your analysis and not use your analysis based on that. But yeah.
What's the longest, like, I guess, sequence you can PCR? Because it doesn't have to be short sequences.
Yeah, it has to be fairly short sequences. It can't do the entire chromosome. And again, this depends on how big your primers are. What was it? There's a couple thousand base pairs that you can do. I'm forgetting the upper limit of it now. Is it 2,000 or 5,000 or I don't know, something like that?
So a mistake every 9,000 base pairs isn't as detrimental.
Yeah, so it's probably not going to be every single PCR product that was made.
Okay.
But on average, it might do 2 PCR products of 1,000 pieces or 1,000 base pairs. Mm-hmm. And so like, every 1 out of every 9 times, there'd be 1 mistake in it. So, all right. Keep repeating, keep repeating, keep repeating. All right, we're going to transition, and now we've talked about replication, making new copies of DNA. We're going to switch now, and we're going to start into transcription. Transcription is when you take a DNA sequence and transcribe it into an RNA sequence. Okay? So if you think of a transcript, if someone took a transcript of our lecture here, they would take English that was spoken and write it down in English that is written. Right? It's the same language, but it's just a slightly different media. Right? Instead of vocal, it's written. That's what's going on here. We're just transcribing the DNA. It's the same language, nucleotide language, but we're just taking it from one form, one media type, DNA, and just same language, but we're just moving it into RNA. Okay, so this is why it's transcription. And this is done by RNA polymerase. RNA polymerases read a DNA template strand, but what they make is an RNA copy. Okay. In prokaryotes, there's usually just one RNA polymerase, and it's going to do all of the transcribing that cell needs. There might be some that have 2. In eukaryotes, you've got a lot of different ones. You've got one that's responsible just for doing the mitochondrial DNA. There's a couple that are for, could be 3 or sometimes more, that will specifically copy certain types of RNA that you want to be made. So there's multiple kinds. There's bacterial kinds, they're unique, viral kinds. There's several eukaryotic types of RNA polymerase. There's ones that are specific for mitochondrial. I'm just going to talk about them all in a very generic form today, just what is the basic function of all of them. And when we actually get to gene expression and start talking about other stuff, we might start talking about the differences between RNA pol 1 versus things that are transcribed by RNA pol 2. But we'll get to that. RNA polymerase is a big multi-subunit complex. And this is a space-filling crystal structure of it. This is where it actually grabs DNA. So RNA polymerase is going to go sit down on double-stranded DNA. And it's going to hold the DNA in this channel that it's got. It's very specific in the sequence that it's going to sit down on. So RNA polymerase will not just go sit down on any region of the genome. It's got very specific sequences that it likes to bind to. DNA polymerases are kind of, well, they're not dumb, but they're generic. Right? You can get a DNA polymerase to sit down anywhere there's a primer. If there's a primer, DNA polymerase will sit down on it and continue to make it. RNA polymerase is not so. It's going to be very sequence specific about where it goes. And that's because you don't want the entire genome made into RNA. Now, I say that, but it turns out that actually most of the genome actually does get turned into RNA. We've been finding more and more that what we thought were just empty regions that didn't have any function of the genome, they actually are being transcribed into RNA, and they do all kinds of regulatory functions that we didn't know before. But again, the RNA polymerases are only going to sit down on certain sequences. It seems like they're doing more than we originally thought, but they are doing it in a sequence-specific manner. Okay, so it binds that double-stranded DNA, and it has got helicase activity. It can pull those 2 strands apart on its own. DNA polymerase can't do that. You have to have a separate helicase enzyme to pull the strands apart in replication. RNA polymerase can do it itself. So it binds double-stranded, pulls it apart, and then it will start polymerizing an RNA copy. It's going to be the reverse complement of whatever it's reading, but it's going to be doing it in the same way that DNA polymerase polymerizes nucleotides. It starts with 5' and it adds new nucleotides on the 3' end. So it's always copying 5' to 3'. Okay? The new thing that's being made, the RNA that's being made 5' to 3'. Now, when it makes that RNA complement, just like DNA polymerase, the RNA, the new strand of RNA, is going to be sticking to the template strand of the DNA that it's reading. Okay? So then RNA polymerase has a second helicase domain. So it's going to open things up in the middle. It's going to start making its RNA strand, and then it will actually pull apart the RNA strand from the template strand so that the RNA piece gets released. We're not just copying the genome. We're just making a temporary copy that we want to go away. Right? So it's got a second helicase activity. All right. Now, I need to step back and talk about chromatin again. Because the DNA is never just naked double-stranded DNA in your cells. Okay? And RNA polymerase has to bind to naked double-stranded DNA. So RNA polymerase does not determine what sequences it gets to stick to. Right? If you've got your genome all wrapped up in histones here, so here's the DNA wrapped around the histone octamers. Here's those are nucleosomes. Those nucleosomes are wrapped around each other by histone H1 into the solenoids. And then the solenoids, again, right, can be stacked and looped and condensed. If you've got condensed DNA, even if you've got DNA just at the nucleosome level, RNA polymerase can't stick to that. It has to have naked DNA. So there's an additional layer of deciding what portions of the genome are accessible to even allowing RNA polymerase to come sit down and make a copy of it. Okay? So a big part of whether or not you're going to make a piece of DNA or not is based on what level of condensation the genome is at. Okay? We've talked about this a little bit before, but this is regulated on many different levels, but at the fundamental level, you add these active groups to the histones in the histone octamers. Okay? So even if I have my portion of the genome in euchromatin, right? It's unraveled pretty much as the genome is in cells. It's still going to be wrapped around by those histone octamers. So if I'm actually going to transcribe that region, it has to be condensed at least to that level, and then I've got to release it from the histones. And the way you do this is you tag those histones by those trailing N-terminal ends that come off of it. So here's another schematic of the nucleosome. So here's DNA, right? 2 strands of DNA colored different colors. And the histones that are making up that octamer are these guys in the different colors inside here. And can you see the little purple guy trailing off here? That's the free N-terminus of one of those histones in the octamer. We can modify that. You come in with enzymes and you stick functional groups on that, and that basically tells the nucleosome either stay condensed or unravel. Okay? So this is called the histone code. And I'm just going to give you general themes about how the histone code works, because we're still actually trying to figure this out. The rules aren't hard and fast, but in general, there's a couple things you can do. You can either add acetyl groups to the histone. The enzyme that does this is called acetylase. Okay? So you have to have a separate enzyme to come in and stick the acetyl groups on. That typically loosens up the DNA from the histone octamer and allows for the RNA polymerase to be able to come in, gain access to the DNA, and actually make you an RNA copy. Okay? If you remove the acetyl groups, and there's a different enzyme for that, it's a deacetylase, that typically codes to tighten it up. Makes it inaccessible for making RNAs. Okay? You can also add methyl groups. Methyl groups tend to repress, but sometimes they don't. And do I have a fourth one? You can also phosphorylate. I don't have it on the slide, but you can also phosphorylate. And there's multiple locations. So you could actually have a couple of places on this N-terminus acetylated, maybe one phosphate, maybe a couple of phosphates, maybe a methyl group. So there's like a code that's there going on somehow. 3 methyls and 2 phosphates indicate a certain level of admittance for RNA to gain access there. And we still haven't quite figured it out. People are still working on it. But we're trying to get a clue on what is the code for really saying that this portion of the genome should be made into RNA. Does that make sense? I know that's a little bit vague, but I just want you to get the idea that it could be inaccessible or accessible depending on how you tag those tails. And I've got 8 tails off of this as well. So there's just one of them. There's another purple one here that you can't see. There's 2 greens, there's 2 yellows, there's 2 reds. Each of those have N-terminal tails, and all of those have the potential to be modified with acetyl groups, methyl groups, and phosphate groups.
What do phosphate groups do again?
They aren't hard and fast, so sometimes one activates, sometimes, and it depends on where you put it as well, because we've got a dozen or more amino acids that you could actually stick these groups on. So suffice it to say, it's kind of complicated, and we don't quite know. It might be a good paper topic if you're interested in that. There's some interesting research there. Okay, that's not the full story, because even if that histone is loose, you still have to have what's called remodeling proteins. Remodeling proteins are going to come in, and if that histone is tagged in such a way that it's loosely associated with the other ones, then a remodeling protein, and there's many, this is a whole class of remodeling proteins, can come in, and they can actually displace the histone octamer, and now I've got some accessible DNA that I could actually make some RNA off of. Okay. I'm just going to introduce this because we're almost out of time. Say I do actually have an accessible piece of DNA. Here is the initiation site for transcription. That's where we actually want to start making RNA. And here is the sequence that the RNA polymerase is actually going to sit down on. RNA polymerase itself can't sit down there. It's got to have more protein helpers to get it down. So hopefully you're getting the theme of how difficult it is and how tightly regulated it is whether you make an RNA piece, right? You could have the chromosome completely condensed in heterochromatin. Or you could unravel it to euchromatin, but it still might not. The histone code determines how tight that is. Remodeling proteins determine if it's accessible at all. And then these additional helper proteins have to come in and help RNA polymerase to actually sit down. So it's no trivial thing to just make a piece of RNA. The cell has got lots of levels of regulation about whether this is going to happen or not, because you don't want to express genes that are inappropriate for the cell type or the cell's environment. So we'll talk about actually how transcription goes, and we may not get to translation before the exam. We'll just have to see. Have a good weekend.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.